We're now going to look at how meiosis differs and how it's similar to mitosis. So just to review, we learned about mitosis and how we go from interphase and then cell division or mitosis, prophase, metaphase, anaphase, telophase, PMAT. And so we're going to start out with a diploid cell and end up with two identical diploid cells. The first thing you need to understand is that when we're going through meiosis, we start out with a diploid cell, but we're going to end up with 4 haploid cells, or 4 cells that only have one set of chromosomes. So let's review this mitosis first from before. Remember, during interphase, during S phase, it's going to make a copy of itself, DNA will, and so that's what each of these sides of the X are. Here's 2 copies, 2 exact copies of the same thing for DNA before the cell divides. You can kind of think of what happens to DNA between interphase and prophase as kind of like a big ball of unwound yarn. And then when it condenses or turns into what looks like X's, that's where it's getting all organized like these skeins here, as opposed to this big mess. When it's duplicating, you can think of it as 1 skein versus 2, where the last place it's attached together is the centromere. So here is a chromosome. The chromosome is made up of 2 identical sister chromatids attached by the centromere. Remember, mitosis is division of the nucleus, and if you remember PMAT and those letters, that should help you. In prophase, the pairs condense, so they're turning into these little bundles or these little X's. We're starting out with a diploid cell, so we have 2 copies of each chromosome. During metaphase, the M stands for middle. They line up in the middle. The last place they're attached here is the centromere, and there's a spindle fiber, these little things like fishing lines, and they have like a tug of war where it attaches to the centromere of each one and pulls them into the middle. And then in anaphase, what's actually happening is the centromere breaks, and each sister chromatid moves to either end. So remember, A for anaphase, A from away from the center. And finally, T for telophase, they're at the 2 ends. If this is an animal cell, you'll see it pinch in and form a cleavage furrow, and we end up with 2 identical diploid daughter cells. So that's what we just went over here. Now we're going to look at how meiosis is different, because we're going to end up with 4 haploid cells at the end of this. So notice we start pretty much the same. Start with interphase. Okay, they're starting to condense and become more organized after it makes a copy. Now, because we're going to be going through 2 phases or 2 reductions, we basically are going to see prophase 1, PMAT with the 1s behind it, and then we'll see PMAT with 2s. So in this first one with prophase 1, something special happened. We have our 2 copies of our chromosome, one from our mom, one from our dad, and each one has its own copy. So what it forms is a tetrad, and tetra means 4. So if you've ever played Tetris, you know that each of the pieces that fall down are combinations of 4 boxes that are put in various configurations. And what happens during this time is something called crossing over. And so during crossing over, parts of one chromosome will break off and reattach to its homologous pair. So we have new combinations and new variations. So this does a good job of showing you one option for crossing over and what happens and how you get lots of different combinations in the offspring. So when forming the gametes, the egg or the sperm, how you can have even more variation in children. Okay, in metaphase 1, our pairs are going to line up. So notice it's not single file, but in pairs, they're lined up in the middle. Each X is going to move to either end in anaphase 1. And then we have the entire X still together at the end of telophase 1. So as we go into meiosis 2, we actually have 2 cells. So prophase 2, the nuclear envelope starts to disappear. Now we see them single filed. They're lined up, and notice they're in 2 cells, not just 1. In anaphase 2, each one is, each sister chromatid breaks, or the centromere breaks, and each sister chromatid goes to either end. And finally, we end up with 4 haploid cells at the end after telophase 2 at cytokinesis. So we have only 1 set of chromosomes, or half the number of chromosomes in this.